Okay, so virus genome is. Do you want any specific
Well,
questions?
one of the questions I had, it said here, identifying and classifying organisms based upon shared features of common scientific practice. And it says, what system or features might you use to be able to organize viruses? Now, I wasn't clear on like what they were talking about there. Are they trying to say like, would you organize them through the Capsids or?
Oh, yeah, that could be one.
Because it's asking us to do our own system on how to classify them, but I didn't know how you would do that.
So you're right. One of them would be the capsids.
Yeah, I was thinking the capsids and also the polypeptide, or not the polypeptides, the DNA, RNA, the
genome.
The genomes that are in it.
Absolutely. Can you think of anything else that could be different among viruses?
The phages.
So host range.
Yeah, the host range.
Host range, what they can infect.
Yeah, and then the only other thing I could think of would be nuclear envelopes, but that falls under capsids, doesn't it?
No, envelopes are different.
Envelopes are different.
Yes, that's the only other thing I can think of.
Yeah.
Envelopes.
Because I kind of put envelopes under capsids.
So what's the difference between an envelope and a capsid?
Well, the capsid is just kind of like the shell that holds the nucleotide in it, and the nucleic acid in it. It holds the nucleic acid in it, and they can be shaped like that, or they're cones, or they're weird.
They can be like cylinders.
Cylinders, yeah. And then the nuclear envelope is part of the host membrane, isn't it?
Yeah, so they can have an envelope. Yeah.
And it takes the glycoproteins from the host cell.
And what's the benefits? Why does this virus do this?
Well, then now it can connect to any host cell it wants to without any problem.
Yeah, it's like camouflage, right? Yeah. Like a white blood cell or a phage that wants to eat bad and foreign bacteria or viruses.
It won't eat it because it'll see them.
Yeah, it'll be like, oh, that's a friendly glycoprotein.
Yeah.
I'll leave you alone. But it doesn't know that there's a virus inside.
Oh,
awesome.
Then one of the other questions it asked about glycoproteins and where is it? The difference between glycolipids and glycoproteins. What's the difference between those two?
They both serve similar functions. The only difference is one is a sugar attached to a protein. One is a sugar attached to the phospholipid.
A phospholipid, okay.
Yeah.
But they pretty much do the same thing then.
Yeah, I mean, if you look on there, I think they're both for cell recognition. I don't remember anything different.
I don't know if it showed up here. It showed up in a different. Say which section did it show up in?
Remember, it's on one of these study guides. It talks about the different.
Yeah, it showed up in the last one.
Oh yeah, membrane structure and fluidity. That's right.
Yeah, see, because it asked for question 12. What are glycolipids and proteins? They're pretty much the same thing, but this one's just a sugar to phospholipid, and this one's sugar to protein.
Yeah.
And what is their purpose on the membrane? They both serve the same purpose, right?
I think so. Is the outline down below the vocabulary? I know it touches on it somewhere. Carbohydrates and membrane. Yeah. Functions. Yeah. So these are functions of both of them.
Cell-to-cell recognition and cell markers. Oh, and protection.
Yeah.
Okay, that's what I thought.
They both do all.
But I was thinking they were different for some reason.
Structurally, but besides that,
okay.
functionally, they're about the same, I think.
And there, for viruses, I think I had everything else. Right here, it talks about size range of viral genomes for a bacteria. It's 300 to 1,000, right? And then for an eukaryote, it's 4 to a couple hundred.
So bacteria, if we're talking about base pairs, I think it's 1 to 10 million.
To 10 million, okay.
The virus, well, let's do eukaryotes. Eukaryotes, they're like, I forget, like 100 million or something like. You know what I mean? It's like it's a lot, 100 million plus.
And then you got what is it freaking called? It starts with an A. I can never pronounce it. The different type of prokaryotes. It's a.
Oh, archaea.
Archaea. There you go.
Yeah. I forget what they're something similar bacteria. I think.
Similar to bacteria.
Yeah, because they both have circular chromosomes.
Oh, okay.
Yeah. And then viruses, they're smallest. They have like 400 to 300 different genes.
Yeah, that's what I was thinking. Okay.
Small. Very small.
They're like nothing. All right, and then
there was a question on the test, remember, about that. So See if you try to get that memorized. 'Cause
Yeah.
it'll help you. I remember I got it wrong. That's why I remember.
The lytic cycle, that's when it injects its DNA and it lyses.